Today we've got Dr. Paul Bayman from the University of Puerto Rico, and he's going to give us a presentation on the parasites that parasitize coffee leaf rust.
Thanks, Matt. Thanks to all of you from for showing up this evening. It's 6 o'clock here in Puerto Rico. Can you see my screen?
Yes, we can.
That's odd. I can't. Give me a second here. There we are. Feel free to interrupt at any time because not everybody may be on the same page with everything I'm talking about. But I want to talk about parasites of coffee leaf rust and how they might be useful for biological control. They haven't been used successfully so far, but I think there's potential there. Yeah, please interrupt at any time if you have any questions. You all know coffee leaf rust, unfortunately. You've all had the bad luck to run into it. Coffee leaf rust has been here in Puerto Rico a lot longer than it's been in Hawaii. It arrived in 1989. It does a fair amount of damage, but people here don't pay as much attention to it as they do as is happening right now in Hawaii. Maybe at first it was a lot of it was more of an issue. But so how can you control coffee leaf rust? Well, resistant cultivars, fungicides like copper or triazoles, cultural control, there's some things that can help, and biological control has been proposed but never really established. There's no real biological control for coffee leaf rust that's available. And there's obviously interest in coming up with something that would work, some organism that attacks coffee leaf rust and helps to control it. Here in Puerto Rico, we have a different situation than you do because a hurricane comes through every now and then and wipes out a lot of the coffee plants. And so as the coffee plants are replaced, more and more they're replaced with resistant cultivars that are resistant to the coffee leaf rust. Limani, Fronton are two local varieties, Obata from Brazil, Marsellesa. A lot of our coffee got wiped out by Hurricane Maria in 2017, and almost all of the coffee that has been planted since then is resistant. So a lot more of our coffee is resistant than in Hawaii. And to tell the truth, most growers don't bother to control it. They don't have the budgets, they don't have the manpower, they don't have the knowledge or infrastructure to do it. So the coffee rust that's there is there, and no one's really sure how much of an effect it makes on yield. It's usually not enough to wipe out the plants, but it certainly affects the yield to some extent. Some people use fungicides. The ones that they use are not necessarily those registered for coffee. So we're interested in biological control, an organism that we can find that would help control coffee leaf rust. And we've been focusing on one, maybe the most obvious enemy of coffee is a fungus called Lacanicilium lacani. It's a hyperparasite. That means it's a parasite of a parasite or a parasite of a pathogen. Here you can see a leaf with the orange coffee leaf rust lesions, and on a couple of the lesions are these white spots. This is a fungus that's growing on top of the coffee leaf rust, attacking it and killing the spore. Lisa Keith is doing a similar project, but she's looking at a much wider range of fungal hyperparasites of coffee leaf rust. There are a lot of organisms that can attack it. We've only been looking at this one, Lacanicilium lacani, And here's a picture of the fungus and its spores. It's a relative of Bavaria bassiana and also of cordyceps, which is the ant zombie fungus, which you've probably heard of. So I'm going to talk about Lecanicilium and some experiments that we've done to see whether it can be effectively used as control. So it's also found in Hawaii, Mexico, Colombia, Peru, probably everywhere where coffee is grown. It not only attacks the coffee leaf rust, it can also attack other fungi and it attacks insects as well. And it's used commercially in biocontrol formulations against other pests and pathogens. It's just never been, there's just no formulation for it for coffee leaf rust. And previous studies on this fungus against coffee leaf rust have been sort of inconsistent. There's never been any really solid evidence that it could be used effectively. So today I want to talk about three questions. How diverse Lecanicilium is, the Lecanicilium that attacks coffee leaf rust in Puerto Rico. Do the different strains work differently? And are there differences in efficacy in different parts of the island and between wet and dry seasons? Is there variation in how well it seems to work? And so the first thing we did is isolated a lot of strains of the fungus from parasitized lesions. Here you can see a lesion. It's easy to pick off the spores under a microscope, grow them in culture, sequence their DNA. Steve Rayner at the USDA in Beltsville has been sequencing our strains, sequencing the ITS and 3 protein coding genes, and has made a number of phylogenetic trees. And what we thought was one genus, Lecanicilium, and one species, Lecanicilium lecani, turns out to be 6 species in 3 different genera. Here you can see the 3 genera on a phylogenetic tree of the family Cordycipitaceae. So it's much more variable than we realized. And maybe this explains part of the inconsistencies of previous studies. They weren't always looking at the same thing. There are a lot of fungi that look the same, and if you don't sequence them, you don't really know who you're dealing with. So we did both lab inoculation experiments and field inoculation experiments. Here's Giovanna Marino during the pandemic, spraying leaves with a suspension of spores of Lecanicilium. And we did experiments where we applied these to coffee leaf rust lesions on coffee leaves. We had 4 treatments. We sprayed spores on leaves with coffee leaf rust and without coffee leaf rust. And we also had controls where we just sprayed water. And so we followed these for 4 weeks to see the effect on coffee leaf rust lesions. And we measured incidence, which is the percent of leaves on each branch that have coffee leaf rust, and also severity, the percent of coffee leaf rust area that's colonized by the parasite. Here you can see a lot of white areas, but you can also see a lot of areas with coffee leaf rust without the fungus. So looking at this just by eye, it looks like what, maybe 40% of the coffee leaf rust area is colonized by the hyperparasite. Any questions so far? Good. Well, so when we inoculate these onto coffee leaves, are they all really hyperparasites? Yes, all the ones we inoculated are hyperparasites, and you can see this is the level of hyperparasitism on coffee leaves after we inoculate 4 different fungi compared with uninoculated controls. You'll notice there are bars here in gray. There is some level of hyperparasites with the uninoculated controls. That's because the fungus is naturally in the environment. You can probably see it on your own farm if you're unlucky enough to have coffee leaf rust. You look at the leaves, some of them will have these white lesions. So we are getting some natural background, but we're also seeing a significant effect of spraying the fungi we applied. Uh, do some of the species work better than others? Definitely. Here we can see one, the second bar, is far higher in incidence after it's being applied than the other three. And here's the uninoculated control. The difference in severity isn't as pronounced. There's still significant differences, but there's more difference in incidence. In other words, how many leaves have the hyperparasite than there is in how much of the rust is colonized. But it varies amongst sites. Here we have in the three different color bars are three different parts of the coffee growing area where we did the experiments. And you can see, for instance, this first fungus is far more efficient in Ciales, sorry, in Lades than it is in the other two sites. And the second one is far more efficient in adjuntas than it is in the other two sites. So each of these sites has a slightly different climate, a slightly different elevation, and that seems to favor which of the fungi will work the best. And we also saw significant variation between the wet season and the dry season. Here, the dry season is January through March. If you look at the green bar, sorry, look at the bars in the pink box. Here's a fungus that in the dry season works much better in adjuntas, pero in the wet season, it works much better in Ciales. It's reversed depending on where you do the experiment. On the other hand, here's one that works about equally well in both wet and dry seasons in both of these sites. But the fungus you choose seems to make a difference, and it has to be adapted to the area where you're applying it. So we think these data are impressive because we see significant differences. They're really able to infect the coffee leaf rust, and this is after only one application. Most biocontrol organisms or any control you use, you would normally apply 2, 3, 4 times in a season. We're seeing an effect after only one application and without any fancy formulations. There's no sticker, there's no protection against desiccation or UV. We just put it in water and spray it, and it has an effect. So we think that this has potential. There is certainly a complication that there's so much variety between sites and seasons. It would be easier if we had one fungus that would work in all areas. And we think maybe one way around that would be to apply a mix of strains and species instead of just spraying one fungus on each plant as we did in our experiments. Spray a mixture of 5 or 6 and hope that one of them is adapted for whatever the weather is going to be at that site. So this is a complication, the variability, but we think that applying a mix of organisms might be a way around it. And it's a much more diverse group than we thought. We found at least 6 species. Some of these have never been reported on coffee leaf rust before, and there are probably more out there that we didn't see. A reminder, whatever control you're looking at, whether it's resistant cultivars or fungicides, the rust has an evolutionary advantage because its life cycle is 3 weeks. Coffee's life cycle is at least 3 years, And the coffee farmer's life cycle is at least 25 years. So the rust can mutate, recombine uh, much more quickly than we can. It can change much more quickly, and that gives it an advantage. We see resistant cultivars or supposedly resistant cultivars with some lesions on them. It's hard to stay ahead of this type of pathogen. I want to take a few minutes to talk about two other aspects of coffee health, uh, mycorrhizae and the coffee leaf miner. Uh, Diana Buitrago is a graduate student here at UPR, a PhD student who's looking at mycorrhizae in coffee seedlings. Mycorrhizae are symbiotic associations between the root of the plant and a fungus that lives in the soil. And all coffee plants are mycorrhizal. Every plant you dig up from your farm will have this sort of association with fungi on the roots because coffee plants can't survive in the field without them. And it's a mutualistic relationship where both partners benefit. The fungus helps the plant gets nutrients, especially phosphorus from the soil. It also helps water uptake and helps protect the plant against disease and nematodes. And it's most important in acid soils because you've probably seen this graph before. This shows pH on the top, and the width of each bar shows the availability of nutrients in the soil at that pH. And if you look at phosphorus, if you get below pH 6, even if there's phosphorus in the soil, it's not available to the plant. And most coffee soils in Hawaii and in Puerto Rico are pretty acid. So mycorrhizae are very important in helping coffee plants get that phosphorus from the soil. And Diana has found so far basically only one fungus in our coffee plants, and she finds significant differences amongst cultivars in the extent of mycorrhizal colonization. And this has important implications for how those plants are going to be able to obtain nutrients and also fight off disease. So this is an area which needs a lot more work, but we hope that one day the World Coffee Research page, in addition to saying for a certain variety, uh, what sort of how resistant it is to leaf rust, what sort of cup quality it has. This page should also be telling you how strongly mycorrhizal this coffee plant is. Uh, that information isn't available yet, but it's a very important part of plant nutrition and plant health, which in coffee has mostly been overlooked. And the other aspect I want to mention very briefly is the coffee leaf miner. Julia was talking about it in her talk last week, which fungicides could be used in Hawaii if coffee leaf miner ever makes it there. This is the larva of a moth that makes tunnels in the leaf, in the mesophyll of the leaf. It leaves both surfaces and eats the middle of the leaf. And here in Puerto Rico, it hasn't been studied much, but it looks to us like leaf miner is far more damaging than coffee leaf rust. Because look at how much of the area of this coffee leaf is useless because of the damage of the larvae. And there are examples that are far more extreme than this one. Here's Giovanna and Luz in the field last week, Giovanna Marino and Luz Serrato, who will be talking later in the series. Here's Giovanna with a leaf with a fair amount of coffee leaf rust. And we found something very interesting when we looked closely at this rust. This lesion, or whatever you call it, this area of damage, actually looks a lot like the face of Puerto Rico's leading export at the moment, Bad Bunny, whose latest album has a song about Hawaii. And if you compare this coffee leaf rust lesion, sorry, this leaf miner lesion, it looks a lot like bad money, at least to the students in the lab, who hope it's a good omen and will help them get tickets in his next series of concerts. So we'd like to thank the people who've helped us in the field, the coffee farmers we work with, the SCRI project, SHAC, and the Puerto Rico Science and Technology Research Trust. And I'd be happy to answer any questions you have.